All right, future orthopedic surgeons, ready to dive deep into muscle physiology.
Always ready.
We're talking the nitty-gritty stuff here,
The kind of stuff that really makes a difference when you're prepping for those big postgraduate exams.
like the FRCS.
Exactly.
And to guide us through this intricate world, we're using the OrthoMade Simple Notes.
From mokyingren.sg,
They're created by Dr. Mok Ying Ren, a sports surgeon, by the way.
who absolutely crushed his FRCS exams back in 2022.
So we know we're good hands.
Absolutely.
Now, I know what you're thinking, more facts to cram.
Right.
But trust me, understanding the why behind how muscles work, it'll make those facts stick.
Yeah. Way better than just rote memorization.
Plus, we'll uncover some pretty surprising insights along the way.
Like, did you know that pulling on a muscle can actually make it contract at first?
Crazy, right?
It's true.
Intrigued.
I hope so.
Let's get started.
Right, let's build a muscle.
So let's pretend we're building a muscle from scratch, like Legos, but with microscopic components.
Okay, I like where you're going with this.
Where do we even begin?
Well, you always start with the smallest building block.
Makes sense.
And in this case, that's the sarcomere.
Sarcomere. So that's like our tiny engine.
It's the fundamental unit of contraction, yeah. Like the engine that drives the whole system.
Got it. Then what? Do we just start stacking them up?
Pretty much. We bundle those sarcomeres together to create myofibrils,
Myofibrils, okay.
which are essentially chains of these contractile units. Think of them as tiny threads within the muscle fiber.
So sarcomeres into myofibrils, got it. But muscle fiber is a single cell, right? How do we get from these tiny threads to, like, an entire muscle?
It's like weaving a tapestry.
Oh, okay.
Those myofibrils are packed within the muscle fiber, and then multiple muscle fibers are grouped together into fascicles, like bundles of threads forming a thicker yarn. Finally, numerous fascicles combine to create the whole muscle that we can see and feel.
So it's like a hierarchy. Sarcomeres to myofibrils to muscle fibers to fascicles, and finally, the whole muscle. Each level builds on the one before it.
Exactly.
And just like each of those Lego creations needs some kind of structure to hold it together, muscles have connective tissue coverings at each level.
Right.
The endomysium around each muscle fiber, perimysium around the fascicles, and epimysium encasing the entire muscle. It's amazing how organized it all is.
It really is.
But we've built our muscle now. How do we make it move?
Good question.
What happens when the brain says, hey, muscle, time to contract?
That's where the magic happens.
Okay, lay it on me.
It's like a beautifully choreographed dance.
I like that.
A 5-step process called the cross-bridge cycle.
Okay, I'm picturing a tiny muscle ballet now.
Perfect.
What's the first move?
It all starts with a signal from the nervous system. An electrical impulse arrives at the neuromuscular junction,
Neuromuscular junction, got it.
releasing a neurotransmitter called acetylcholine. This triggers a wave of electrical activity that spreads across the muscle fiber membrane.
Okay, so the signal is received. What's next in this muscle dance?
Calcium takes center stage. That electrical signal causes the sarcoplasmic reticulum, which is a storage network within the muscle fiber, to release calcium ions.
Calcium, why is that so important? What does it actually do?
Think of calcium as the key that unlocks the contraction process. On the actin filament.
Actin, those are the thin strands, right?
Yeah, within the sarcomere.
Okay,
There are binding sites where the myosin heads, the thicker filaments, want to latch on.
so the myosin wants to grab the actin but can't. What's stopping them?
Exactly. The binding sites are blocked by a protein called tropomyosin. It's like a bouncer at a club, keeping the myosin heads out.
Okay, so how do we get past the bouncer?
Calcium comes in. It binds to another protein complex called troponin, which is attached to tropomyosin.
Troponin, right.
This binding causes tropomyosin to shift, revealing those hidden binding sites.
So calcium is like paying off the bouncer to let the myosin heads in.
Basically.
What happens next, now that they're in the club?
Now the party really starts.
Okay.
Now that the binding sites are exposed, the myosin heads can attach to the actin filaments.
Okay, got it.
This is where the sliding filament theory comes into play.
Okay, sliding filament theory, I've heard that term.
Yeah.
But honestly, what's actually sliding?
It's the actin and myosin filaments themselves.
Oh, okay.
Imagine this. The myosin heads attach to the actin, pull the actin filaments inwards, then detach, reattach further down, and pull again.
So they're like crawling along?
Kinda, it's like a microscopic tug of war.
That makes sense.
This repeated grab and pull is called the power stroke, and it's what causes the entire muscle to shorten.
Wow. It's like a perfectly coordinated microscopic machine.
It really is.
But to really get this, I feel like we need to zoom in even further.
Okay.
Explore the sarcomere itself. What are we looking at under a super high-powered microscope?
When you magnify a sarcomere, you see these alternating light and dark bands
Oh, yeah?
called striations. They're created by the arrangement of those thick myosin and thin actin filaments we've been talking about.
So those striations are what gives skeletal muscle its striped appearance.
Precisely. And within those striations, there are some key landmarks you need to know.
Okay, hit me with it.
First, we have the I band and the A band.
Those sound vaguely familiar, but refresh my memory.
Sure.
What do those bands represent?
Think of it this way. I stands for isotropic, meaning the I bands look uniform under a microscope because they only contain the thin actin filaments.
Nelly Acton. Okay.
A stands for anisotropic, meaning the A bands look less uniform because they have both the thick myosin and thin actin filaments overlapping.
So the I band is light and uniform,
Yes.
while the A band is dark and a bit more jumbled because of the overlapping filaments.
You got it.
Are there any other important landmarks within the sarcomere?
Absolutely.
Tell me more.
There's the Z line, which marks the boundaries of each sarcomere. It's like the dividing line between two houses on a street.
Okay, like a fence.
Exactly. And then there's the M line, which runs down the center of the sarcomere,
M line right down the middle.
right in the middle of the A band.
So we have these zones and lines within the sarcomere. Can you give me a quick rundown of where they all are?
Sure, picture this.
Alrighty.
The Z line is at each end of the sarcomere. The I band is the lighter area on either side of the Z line containing only actin. Then we have the A band, which spans the central region and includes both actin and myosin. And finally, the M line is that thin line right down the middle of the A band.
Okay, I think I could draw a sarcomere right now and label all the parts.
I bet you could.
But let's shift gears a bit. We've talked about how muscles contract, but is it simply an on or off switch? Does a muscle always contract with the same force?
That's a great question. It leads us to a crucial concept: the relationship between muscle length and the force it can generate.
Oh, I remember this.
This is described by the Frank-Starling curve.
Okay, Frank-Starling curve. What's the main takeaway here for someone who's, going to be treating muscles in the real world?
The force a muscle produces, it depends on how much the actin and myosin filaments overlap.
All right.
There's a sweet spot, an optimal length,
Yeah.
where you have the maximum number of potential cross bridges
Cross bridges.
leading to the greatest force generation.
So if a muscle is too stretched out or too bunched up, it can't generate as much force.
Exactly. Imagine trying to shake someone's hand with your arm fully extended or completely bent. You can't generate much force in either of those positions.
Yeah, that makes perfect sense. But wait, there's another law that relates to this, right?
You're right.
The law of von Schwann.
Yes, the law of von Schwann states that muscle tension actually decreases as the muscle fibers shorten beyond a certain point.
Wait, really?
It might sound counterintuitive, but think about it. When those sarcomeres get too short, the filaments can't slide past each other effectively. They're basically getting in their own way.
Interesting. So there's this delicate balance between filament overlap and shortening. Too much or too little of either, and the force drops. But how do the Frank-Starling law and the law of von Schwann work together?
That's where it gets really interesting.
They do it.
And where understanding these principles becomes crucial for things like rehabilitation.
Of course. So important.
We'll dive deeper into that after a quick reminder that all this information is based on the Ortho Made Simple notes from mokingren.sg.
Such a great resource.
It's a fantastic resource for anyone studying for those postgraduate orthopedic exams.
Back in a moment.
See you soon! Welcome back. Before the break, we were discussing that fascinating interplay between the Frank-Starling law and the law of von Schwann.
Right, and I'm still trying to wrap my head around how these seemingly contradictory principles actually work together.
Yeah.
On the one hand, you've got the Frank-Starling law saying more stretch equals more force.
Right.
But then von Schwann throws a wrench in the works and says too much shortening actually reduces the force. So how does the muscle find that sweet spot for optimal contraction?
That's a great question. And it gets to the heart of muscle physiology. It's all about finding that balance between those two forces.
Okay.
Think of it like a rubber band.
Oh, yeah, rubber band.
If it's too slack, you can't generate much force, but stretch it too far, and it could snap muscles work in a similar way, constantly adjusting their length and tension to achieve the desired movement.
Okay, that analogy makes sense. But how does this apply to real-world scenarios?
Yeah.
I mean, we're not just stretching rubber bands as orthopedic surgeons, right?
Absolutely. Understanding this interplay is crucial for everything from diagnosing muscle injuries to developing effective rehab programs.
Okay.
For example, if a patient comes in with a hamstring strain, knowing the optimal length-tension relationship helps us determine the appropriate range of motion for exercises and stretches.
So instead of just blindly prescribing a set of exercises, we're using this knowledge to tailor the treatment to the individual patient and their specific injury.
Exactly. It's about working with the muscle's natural physiology to promote healing and restore function.
And speaking of function,
Yeah.
let's talk about those different types of muscle contractions.
Okay.
They're not just textbook terms. They have like real implications for how we train and rehab muscles.
Absolutely.
Okay, bring on the contraction variations. I remember learning about isotonic, isometric, and a few others, but honestly, it's all a bit hazy. Can you break it down for me?
Of course. Let's start with isotonic contractions, which are probably the most familiar.
Probably, yeah.
Isotonic means the muscle changes length while maintaining a constant tension.
Okay.
Think about lifting a dumbbell during a bicep curl.
Classic.
As you lift the weight, your biceps shorten, performing a concentric contraction. Then as you lower the weight, your biceps lengthen while still controlling the movement, performing an eccentric contraction.
Right, so isotonic means the tension stays relatively constant, but the muscle length changes. Concentric is shortening, and eccentric is lengthening. I'm starting to remember this now.
Good.
What about isometric contractions?
In an isometric contraction, the muscle length stays the same, but the tension increases.
Okay.
Think about pushing against an immovable object.
Like a wall.
Like a...
You're generating force, but there's no movement.
Exactly.
Like planking.
Yeah.
You're working those muscles,
Yeah.
but you're not moving any joints.
Right.
And what about isokinetic contractions? Those always sounded kind of futuristic to me.
Isokinetic contractions require specialized equipment
Oh, okay.
that controls the speed of movement, providing constant resistance throughout the range of motion.
Interesting.
They're often used in research and rehab settings
Yeah.
to assess muscle function and strength.
So we've got isotonic, isometric, and isokinetic, each with its own characteristics.
Right.
But how do these different types of contractions factor into rehabilitation? Do we use all of them for every injury?
That's a good question. The choice of contractions really depends on the specific injury, the stage of healing, and the patient's goals.
Of course.
For example, in the early stages of rehab, we might focus on isometric exercises
Okay.
to activate the muscles without putting too much stress on the injured tissue.
Makes sense, like easing the muscle back into action.
Exactly. As the muscle gains strength, we can incorporate concentric and eccentric exercises to build strength and restore full range of motion.
That makes sense. So it's all about tailoring the exercises to the specific needs of the patient and the stage of their recovery.
Yeah.
But there's another distinction I keep hearing about. Open chain versus closed chain exercises.
Right.
Are those just fancy terms for different types of contractions?
Not exactly. Open chain and closed chain refer to whether the distal end of the limb is free to move or fixed during the exercise.
Okay.
In an open chain exercise, the end of the limb is free.
Yeah.
Think about doing a bicep curl or leg extension.
Okay, so open chain means the hand or foot is moving freely in space.
Right.
What about closed chain?
In a closed chain exercise, the end of the limb is fixed, usually against a stable surface.
Okay.
Squats and push-ups are classic examples.
So closed chain means the feet or hands are in contact with the ground or a solid object.
Right.
Why is this distinction important? Especially for, orthopedic residents prepping for their exams.
Understanding the difference between open and closed chain exercises is important because they place different demands on the muscles and joints.
Okay.
Closed chain exercises tend to involve more muscle groups working together, promoting stability and functional movement patterns. Open chain exercises, on the other hand, allow for more isolated strengthening of specific muscles.
So closed chain for stability and functional movement, and open chain for targeted strength training.
You got it.
Sounds like both have their place in a well-rounded rehab program.
Absolutely.
And this brings us to another important topic for those upcoming exams. Muscle injuries.
Oh, yeah.
Where do muscles tend to be most vulnerable, and how do they heal?
Muscles are remarkably resilient, but they do have a weak point,
Oh, no.
the myotendinous junction.
Myotendinous junction.
Where the muscle fibers meet the tendon. It's like the seam between two different materials prone to tearing under stress.
Why is the myotendinous junction so susceptible to injury? Is it just bad luck, Or is there something about its structure that makes it more vulnerable?
It's a combination of factors, but it's particularly vulnerable during high-velocity eccentric contractions.
Oh, okay.
Think about a sprinter who suddenly feels that sharp pain in their hamstring.
Oh, yeah. I can picture it.
That's often a classic example of a myotendinous junction injury.
Ouch. I can practically feel that pain just imagining it. So what happens when a muscle tears at that junction?
Yeah.
How does it repair itself?
The body's amazing healing mechanisms kick in, but it's not a perfect process.
No.
Scar tissue forms to bridge the gap,
Okay.
but it's not as strong or flexible as the original muscle tissue.
So scar tissue is like a patch job, but it's not quite the same as the original fabric.
Right.
Can this scar tissue cause problems down the road?
It certainly can.
Oh, no.
Scar tissue can restrict movement, reduce flexibility, and even increase the risk of re-injury.
Wow.
That's why proper rehabilitation after a muscle injury is so crucial. It's all about minimizing scar tissue formation and restoring function as much as possible.
Speaking of rehab,
Ah,
I'm reminded of that mind-boggling fact we talked about earlier. Applying traction can actually make muscles contract initially. That seems so counterintuitive. I thought traction was supposed to relax muscles.
it's one of those things that makes you go, wait, what? But it all boils down to a reflex mechanism called the monosynaptic reflex.
Monosynaptic reflex. Okay, break that down for me.
It's a simple reflex arc where a sensory neuron detects a stretch in the muscle and sends a signal directly to a motor neuron in the spinal cord.
Hmm.
That motor neuron then sends a signal back to the muscle, causing it to contract.
So it's like the muscle's knee-jerk reaction to being stretched.
Yeah.
It's saying, hey, I'm being pulled, I better tighten up. But if that's the case, how does traction ultimately achieve muscle relaxation? I mean, we know it's used to reduce fractures and dislocations.
That's where another fascinating mechanism comes into play, the Golgi tendon organs.
Golgi tendon organs, okay.
These are sensory receptors located in the tendons
Okay.
that detect changes in tension.
Mm-hmm.
When a muscle is stretched for a prolonged period, the Golgi tendon organs trigger a different reflex called the Golgi tendon stretch reflex, which actually inhibits muscle contraction.
So it's like a two-step process.
Yeah.
Initial contraction followed by relaxation.
Exactly. The initial contraction is a quick protective response.
Makes sense.
But as the traction is maintained, the Golgi tendon organs step in and say, okay, it's time to chill out now. We don't want to overstretch and cause more damage.
It's incredible how these different reflexes work together to fine-tune muscle function. I'm starting to see how understanding these seemingly small details can have big implications for orthopedic practice.
Oh.
And this understanding is essential for those preparing for their postgraduate orthopedic exams.
Right.
Remember, all this information is based on the OrthoMade Simple Notes from mokingren.sg,
Yeah, a great resource.
created by Dr. Mokingren, Ren, who aced his own exams back in 2022. They're a fantastic resource for anyone looking to simplify these complex concepts.
Definitely.
We'll be back after a short break to continue our exploration of muscle physiology. We're back, ready to wrap up our muscle physiology deep dive.
Definitely.
It's been quite a journey, hasn't it?
From those tiny sarcomeres all the way to understanding how whole muscles work, how they respond to injury.
Exactly. So let's put it all together. Imagine a patient comes in with a classic hamstring strain.
Okay, yeah.
How does everything we've talked about, the Frank Starling curve, the different types of contractions, those Golgi tendon organs, how does all that knowledge guide your approach to rehab?
Well, first step's a thorough assessment, right?
Of course.
We need to pinpoint the severity and location of the injury, which might involve imaging studies. But knowing the myotendinous junction's vulnerability to those high-velocity eccentric contractions, often seen in hamstring strains,
Right.
it gives us a starting point.
So understanding the mechanism of injury is key.
Yeah, it's like knowing how the machine broke down before you try to fix it.
Exactly.
In the early stages, we focus on protecting the injured muscle, managing pain and inflammation. But as healing progresses, we shift our attention to restoring proper muscle function. That's where understanding those different types of contractions
Right.
becomes crucial.
Okay, so how do you decide which type of contraction to use and when? Is there like a specific sequence you follow?
It's not a one-size-fits-all approach. We tailor the rehab program to the individual patient and their specific injury. But generally, we might start with gentle isometric exercises where the muscle contracts without changing length.
Okay.
This helps to increase blood flow to the area and activate those muscle fibers without putting too much stress on the healing tissue.
Makes sense. It's like easing the muscle back into action.
Exactly. As the muscle gains strength, we can progress to concentric exercises, focusing on shortening the muscle.
Mm-hmm.
Think of those classic hamstring curls, gradually increasing the resistance.
And what about eccentric exercises? Those seem a bit trickier, given their role in hamstring injuries in the first place.
You're right, they require careful progression, but they're essential for building functional strength and preventing re-injury. We might incorporate exercises like slowly lowering the weight during a hamstring curl or walking downhill, which emphasizes that eccentric control.
So it's like retraining the muscle to handle those lengthening forces safely, kind of like exposure therapy, but for muscles.
That's a great analogy. And remember the Frank Starling curve.
Of course.
We always need to consider the muscle's optimal length for force generation when designing these exercises.
Right, too short or too long,
Mhm.
and you're not getting the most out of those contractions. It's all about finding that sweet spot.
Exactly. And we also need to consider the patient's individual needs and goals.
Mhm.
A marathon runner will have different rehab goals than a powerlifter.
Of course, makes sense. And I imagine understanding the different muscle fiber types is crucial here as well, right?
Absolutely. We might tailor the training program to emphasize either endurance or power, depending on the muscle involved and the demands of the patient's activity.
Wow, it's amazing how all these pieces of the puzzle fit together. It's like we've gone from building a muscle brick by brick to understanding how to put it back together after it's been injured.
It's a testament to the complexity and brilliance of the human body.
It really is.
And for those of you preparing for your postgraduate exams, remember that understanding these principles is key to becoming a successful orthopedic surgeon.
Couldn't agree more. And before we sign off, I want to give another shout out to those Ortho Made Simple notes from mokiandgren.sg.
Great resource.
They're a fantastic resource for anyone studying for those challenging exams.
Dr. Mok-Ying Ren, who created those notes, clearly has a knack for simplifying these complex concepts.
He really does. So, to all our listeners out there, keep those brains firing, keep those questions coming, and most importantly, keep diving deep into the fascinating world of orthopedics. Thanks for joining us on this deep dive. We'll catch you on the next one.